What we're trying to accomplish in this part of the training of people for biomedical techniques and the skills, one of the most important one of those skills is to be able to take a sample of a particular bacteria from whatever source and then go in and identify what it is by looking at it under the microscope. So every time you go to the hospital, they take a swab of your throat, somebody's going to go down and put it onto a microscope and try to identify it.
These are the professional slides that you would use in a professional environment.
What we're doing is we have plates with different bacteria in them, and we're using we're putting the bacteria onto slides, and we're doing a gram stain so we can look under a microscope and we can tell like if it's gram positive or gram negative. And it's really like kind of cool to look under a microscope with it because we're able to like see clumps and like trends in the bacteria and see like it spread out so we can identify if it's like a gram positive or a gram negative.
Thank you so much. I just put my bacteria using my loop onto the slides with water, and now I'm just making sure that they are staying on the slide by applying heat to it. And then after it gets nice and hot, as you can see the spots, they start to dry up, and then the bacteria is stuck to the slide and it can't move.
When these kids go through this class, they're going to be trained, they're going to know exactly what to do when they go into a laboratory and somebody tells them, I need you to do a gram stain and I need you to be able to do this diagnostic for me. We give them those abilities to do it like professionals. So what we have here is an example from one of my students. This is a really great slide, and she has been able to get the individual bacteria where you can see them very clearly, separate from each other, and it's not easy. You have to be careful not to put too many bacteria onto the slide. You have to get it into just the right orientation, and then the nice thing about this is as you look at these bacteria, you're going to be able to identify what type of bacteria it is. This is a gram-negative bacteria. This would tell a doctor, I can't give them penicillin or ampicillin, it's not going to help. I have to give them a different type of an antibiotic. So these are the kind of decisions that you can make simply by looking at the cells. And so that's one of the reasons why here in the CTE we spend a fair amount of time trying to teach the kids how to do these classical laboratory methods.
So currently we're dyeing our different bacterias for the gram stain.
Okay, how do you do that?
So first you have to dip it in the purple crystal solution. You do it for a minute intervals on each. You rinse it, then you dip it in iodine for a minute, you rinse it. You put a couple drops of decolorizer and then you seal it all in the last, and then it's all in 1 minute intervals.
Right now, I finished my gram stain, so I stuck it in the solutions over there, like the iodine, and now I am on the microscope and I'm looking to just like get it in the frame for now, and then I'm going to use the oil for the oil immersion lens. And what I'm going to do is I'm going to place the oil on top of one of the gram stains, and I'm going to go into this lens right here, and I'm going to be able to like see the spread of the bacteria and be able to figure it out if it's gram positive or gram negative, so like the color will determine that.
So gram positive would be the blue, and what that means is that the blue stain gets stuck into the cell wall. In the gram negatives, it can be washed away, and then you counter stain with a red dye, right? So the red, otherwise it'd be invisible. If it's invisible, you can't really see it, right? So that becomes diagnostic for how the cell wall is built, and that again goes back to how someone would approach it for doing antibiotic therapy if they found it.
Okay, so in this first one, it's a gram negative stain, my first one. This is a gram positive stain. This is another more congested gram positive stain.
like it prepares us for what we could do in the future. So again, with the collaborating and then the hands-on, it's getting us ready for like future jobs and like it actually being in a lab, because we're doing a lot of independent work too. So we're doing stuff on our own. He'll give us directions and we'll just go into it and do it ourselves. So like if we make mistakes, we can try to fix them ourselves like we were in the real world.
I really like the hands-on part of this class. You know, it's like really different to any other science class I've taken. Very hands-on, you're working in a lab and you're collaborating a lot more. It's not a lot of like textbook work or like researching. Well, it is, but mostly hands-on and working in the lab, which I really enjoy.